Hello, everyone. In this chapter, we are going to cover the chemistry of life. As we are going through the chemistry of life, I would like you to focus on the following questions. The first question I would like you to focus on is how are living things organized from atoms to molecules? The second question it's going to be what is pH and how is it important for living organisms? The third question, what are the four macromolecules found in living systems? What are the structures or the building blocks or subunits? And what are the functions of these macromolecules of life? Then the next question is going to be how are proteins organized and how is their shape important for their function? Finally, what is DNA? What is RNA? How are these two molecules different? And what is similar between them? Now, the first step we are going to make is to define matter. Matter is anything that has a mass and takes up space. Anything that is visible, anything that you can touch is matter. That matter is made up of elements: oxygen, iron, carbon, sodium, potassium are examples of elements. These elements are themselves made up of small building blocks that are called atoms. These atoms for a given element are the smallest unit of that element that still retain all the physical and chemical properties of that element. In other words, if an atom somehow is broken into pieces, then the element that atom was representing does not exist anymore. All right? Then the atoms of elements are going to come together to form molecules. These molecules are going to form very complex structures that cause you to exist. Now, let's dive a little more in atoms, basically their structures. Now, before we dive in atoms and their structure, let me first say that in nature, there are about 96 naturally occurring elements. This element can be organized on the Mendeleev table or the periodic table. I'm not going to go into the intricacies of that table. However, some of the information I want you to remember, it's the atomic symbol. Every single atom in the periodic table has a symbol. For instance, nitrogen is symbolized N, carbon is symbolized C. Every element also has what we call an atomic mass. That atomic mass on this table is at the very bottom, right here. For carbon, for instance, the atomic mass is 12. For nitrogen, the atomic mass is 14. Then, but not least, you have the atomic number. The atomic number is going to be the number of a particular type of subatomic particle that we call protons. That we call protons. We are going to see this in a minute. And that atomic number define what element a given atom is going to belong to. For instance, the atomic number for carbon is 6, while the atomic number for nitrogen is 7. The atomic number for oxygen is 8. So anytime you would see an atom with an atomic number 8, it has to be oxygen. If the atomic number is, say, 15, like right here, that atom has to belong to the element phosphorus, right? Now, let's look inside of the atom. An atom is organized in such a way that you have a central component that we call the nucleus, and around the nucleus are gravitating additional elements. Now, in the nucleus of an atom, you are going to have two types of subatomic particles: protons that are electrically positively charged, neutrons that do not carry an electric charge. This is part of what we call the nucleus, the central part of an atom, right? The mass of an atom is that of the protons and the neutrons. And then around the nucleus, we are going to have electrons that are going to gravitate. These electrons typically are going to carry a negative electric charge. So, to summarize, an atom, it's a nucleus made up of protons and neutrons around which electrons are spinning, right? Now, we are going to have electrons spinning around the nucleus based on the amount of energy this electron has. But I'm not going to bug you with the intricacies of that. I want you to know that where an electron is sitting outside of the nucleus, it's called an electron shell, right? Now, based on the element, the number of electron shell is going to vary, right? For instance, oxygen has only one electron shell, while carbon has two electron shells. So we have one here and the second one here. All right? Each electron shell can carry a maximum number of electrons. The innermost electron shell in a given electron can carry up to 2 electrons, never more than 2. While the second electron shell can carry up to 8 electrons, never more. It can carry less, but it cannot carry more, right? So this is an information you want to know. And the reason why you want to know that information is because we mentioned earlier that atoms come together to form molecules. But the question we may ask is what triggers them to come together in order to form molecules? That's a question I want you to keep in mind. Before we address that question though, we notice that some elements, in spite of their atomic number that never changes, we see that this element occur in multiple forms. That's the case of carbon element. Carbon element occurs in 3 forms. We have carbon 12, carbon 13, and carbon 14. Carbon 12 has 6 electrons. It has 6 neutrons. And it has 6 protons. The mass of an atom is that of the neutrons and the protons. 6 plus 6 is 12. Now, let's take a look at carbon 13. Carbon 13, however, has 6 electrons, 6 protons, but it has 7 neutrons. So that together we have 13, which is this number that we see here. Finally, carbon 14 has 6 electrons, 6 protons, but 8 neutrons. Together, we have 14. So, these different atoms that have the exact same atomic number, which is 6, right? Nevertheless, these different atoms have a different mass. That mass is different because of the number of because of the number of neutrons, because of the number of neutrons that vary from one atom to another atom. These atoms actually are called isotopes. They are isotope because they are variant form of the same element, right? The same atom element. Some of the isotopes, in the case, for instance, of carbon 14 actually decays over time. It releases energy. Those isotopes that release energy are called radioisotopes. These radioisotopes can be harmful. They can cause cancer. They can cause cancer and destroy cells. But they can also be very useful. In medicine today, we use more and more what we call nuclear medicine, where we are using radioisotopes in very, very tiny amount for diagnosis purposes to see in the body which part is properly functional and which part is not functioning properly, right? We use radioisotopes to do this, right? And we also use radioisotopes to achieve a number of other purposes, but I'm not going to dive into those details. So remember that the radiations released by radioisotopes can be harmful. How are they harmful? By damaging DNA. Okay, I want you to remember that detail. Now, we use some isotopes, as I mentioned earlier, for diagnosis in medicine. For instance, here, we do have the imaging of a human thyroid. So it is possible to give a patient a solution containing iodine 131. Iodine 131, which is a radioisotope. That radioisotope, in a very small amount, can attach to functional part of the thyroid because they produce the thyroid hormone. And when you provide that isotope, the cells do not distinguish between the radioactive isotope and the non-radioactive isotope. They don't distinguish that because the atomic number is the same. Therefore, iodine is iodine, regardless of if it decays or not. Nevertheless, we are going to use the radioisotope as a tracer so that in this little corner right here, we see that we are missing portion of the thyroid gland. We are missing that portion because the patient has a cancer. And because the patient has a cancer and the cancer cells are not doing the job they are supposed to do in the body, therefore, we are going to see lack of function, which shows that, oops, this patient has an issue, right? Then you have another technology, which is the tomography positron machine that is basically a scanner that you can feed a patient with minute amount of radioactivity in a solution. They drink it, and that solution is going to go and attach to active parts of the brain. So here, a patient with Alzheimer's disease, we are going to see where in the brain you have activity. The red zones right here, surrounded with green, are the areas where activity is happening, so that we can track in an individual who is either prone to develop Alzheimer or is already having Alzheimer. We can track that Alzheimer disease if it's progressing over time or not. So isotopes are very useful that way. Now, atoms are going to come together to form molecules. Molecules are going to be made up of sometimes the same kind of atoms. For instance, oxygen and oxygen come together to form oxygen gas. Hydrogen and hydrogen come together to form hydrogen gas. However, these molecules can also form from different atoms. For instance, an oxygen atom can interact with hydrogen atoms so that you are going to have water. And whenever molecules form out of different kind of atoms, that molecule is called a compound. Compound. Now, how do atoms come together to form molecules? There are different ways they come together to form molecules. The first type of way they come together to form molecules is by forming ionic bonds. In ionic bonds, if we take the example of table salt that we call sodium chloride forms out of two atoms, the sodium atom and the chloride atom. Now, notice that the outermost electron shell for the sodium atom has only one electron, while the outermost electron shell for the chloride atom has 7 electrons. That chloride atom is missing 1 electron. To make things simple here, what happens is that for the sodium to become stable, it's simply going to give away its single electron in the outermost electron shell, while chlorine atom is going to receive an electron. At the end of the day, because sodium has given away his electron, now overall, the chemical now is going to be charged positively. Now we have more protons than electrons in sodium. So that now we don't call it sodium atom anymore. We call it sodium ion because it is charged. Atoms are not electrically charged. The chloride The chlorine atom, on the other hand, that has gained an electron, has gained a negative charge. That's why it is charged here. So now we don't have an atom of chlorine anymore. We do have a chloride ion. So now we have two ions that come together, form crystals, and form the table salt, the sodium chloride salt. So that's one way of forming forming molecules with ionic bonds. The next way to form molecules is going to be by covalent bonds. In covalent bonds, atoms are going to literally share electrons, right? Oxygen, for instance, in its outermost electron shell is going to have 6 electrons. 1, 2, 3, 4, 5, 6. Now, the maximum number of electrons that the outermost electron shell in oxygen can obtain is 8. This element is in need to develop two connections with other atoms so that the outermost electron shell can be stable. Because of that, oxygen will mingle with hydrogen to hopefully become stable by filling their outermost electron shell. When they come together in H2O, a water molecule, we have that the oxygen molecule is now stable because it now has 1, 2, 3, 4, 5, 6, 7, 8. So that electron shell is stable. Now, for hydrogen, the story is the same. The outermost electron shell for hydrogen needs 2 electrons. Now that hydrogen has combined with oxygen, now we have 1, 2. So now hydrogen is stable and another hydrogen here is going to be stable. And this is how the molecule of water gets formed. Let's take a look at oxygen gas. In oxygen gas, 2 atoms of oxygen, one here and another one here, are going to come together by sharing their electrons here so that the outermost electron shell can become stable. So these are covalent bonds. Now, we talked about water earlier, how oxygen and hydrogen come together to form a molecule. There is more to that story than simply forming molecules. We noticed that water is the solvent of life. Everywhere you go, you find water. In your cells, most of your weight is occupied by water, right? Water dissolves everything, right? Or almost. Water is liquid, it can be solid, it can be gas, right? But in its liquid form, water is going to be liquid at room temperature, right? It is not easy for water to change temperature quickly. It takes a while. It takes a lot of energy for water to change temperature, right? Water has a high heat of evaporation, which means that you need to increase the temperature by a lot to actually cause evaporation of water. Frozen water is less dense than liquid water, which causes frozen water to float on top of liquid water. The molecule of water can cling together and form drops of water. Question is, how do they cling together? What causes them to cling together? Now, know that in the relationship where oxygen and hydrogen share electrons, I want you to know and remember that that sharing is not equal. In other words, let us go back here a little bit. In this molecule of water, while there is a share of electron, what is interesting, however, it's that the electrons that are shared here between oxygen and hydrogen, these electrons tend to spend more time close to oxygen than they do to hydrogen. This happens in here, in this relationship. This also happens in this relationship. This becomes very important because the electrons spending so much time close to oxygen, oxygen has now some kind of slight modification so that we have an extremity of oxygen atom that is slightly negatively charged because of the influence of the electron, while on the side of hydrogen, hydrogen is going to be slightly positively charged because of the slight loss. Now, I'm saying slightly charged, positively for hydrogen and slightly charged negatively for oxygen, which causes water to be a polar molecule. Now we have two poles. One pole toward oxygen that is slightly negatively charged, and one pole toward hydrogen that is slightly positively charged. This causes water molecules, as they come together in a glass, to interact based on these slight charges, so that the water molecules are going to cling with each other. This clinging is actually what we call hydrogen bonds. This hydrogen bonds allows water to behave like magnets, attracting each other. That property of water to cling together is also one of the reasons why water is quick to dissolve all other polar molecules. Think about sugar. When you take table sugar, you put it in the water, that table sugar after a certain time is going to dissolve. If you take sodium chloride, which is table salt, you put it in water, that table salt is going to dissolve, right? This is made possible because water is a polar molecule. It's going to dissolve a lot of different chemicals. So I want you to remember the hydrogen bonds. So water is not going to only form hydrogen bonds among themselves, but they are also going to form hydrogen bonds with other chemicals, including sugars, proteins, to name only those, right? Now, I want you to remember that these hydrogen bonds are relatively weak. Now, I may say that they are relatively weak. That doesn't mean that they are absolutely easy to break. I'm not saying that, right? Because when we look, for instance, at a molecule of DNA, where we have a double helix molecule that is stabilized by hydrogen bond, we notice that It takes a lot of energy to break apart these two molecules that are connected together through hydrogen bonds. But that's another story. Let's close that. Let's close that parenthesis. Water molecules can split. They can split, and they can also cause a dissociation of other molecules when we are in a water-based environment. When water molecule splits, so we have oxygen and hydrogen. When water molecule splits, now we're going to have a hydrogen ion and an hydroxide ion. Now remember that in the relationship oxygen-hydrogen, we said that at the end of the day, water is a polar molecule, that oxygen is slightly negatively charged while hydrogen is slightly positively charged. When water splits, now the loss of electron is going to be effective. Oxygen is going to steal the electron from hydrogen that has now lost it. That's why we have hydrogen ion that is positively charged, hydroxide ion that is negatively charged. With that being said, it is possible to quantify in a water-based solution the concentration of hydrogen ion and the concentration of hydroxide ion in a solution. So we can determine that, and rather than going back and forth between concentrations of hydrogen and concentrations of hydroxide ion, chemists and biologists have made things simple. They came up with what they call pH. pH is basically what we call potential. Oops, this is not correct. Potential hydrogen. So we are going to ask the following question. In a given solution that is water-based, what is potentially the concentration of hydrogen? Because of that, we are going to come with a scale. I spare you the details. Now, we notice that some substances, when they are in a water-based environment, these substances are going to release hydrogen ions. The substances that release hydrogen ions, we call them acids. While other substances in a water-based environment are going to rather steal hydrogen ion or release hydroxide ions. These substances are said to be bases. I want you to know the definition of what an acid is and the definition of what a base is, right? An acid substance is a substance that releases hydrogen ion, while a base substance is a substance that takes up hydrogen ion or releases hydroxide ion. Now, with that said, please always keep in mind the potential concentration of hydrogen in a solution. With that said, now, we came with using some math, a way to determine the concentration of hydrogen in a given solution, right? So that at the end of the day, we come with the conclusion that pH is going to be the negative log of a concentration of hydrogen we can be 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, to mention only those. Now, rather than dealing with all the logs, we make things simple. We say the pH, potential hydrogen, for a given solution is going to be simply this value, and this value. So we're going to say pH of the negative log of 10 to the minus 1, it's simply 1. pH, which is equal the negative log of 10 to the power minus 2, it's simply 2. So we have a scale of pH that varies from 0 to 14, with 7 being a neutral pH. Anything below 7 is considered acidic, while anything above 7 is considered basic. So in this course, I'm not going to ask you to compute or to run some equations to determine the pH. That is not the purpose of this course. I want you to know what pH is and how it applies to living organism. We are going to see that in a minute. So when we consider the scale of pH that varies from 0 to 14, 0 being extremely acidic, an example of very highly acidic compound that we know of is hydrochloric acid, while an example of substance that is very basic, it's sodium hydroxide. In between, you are going to have a number of familiar situations. For instance, you use lemon juice. Lemon juice is acidic. The pH is around 2, which means that the concentration of hydrogen ion, it's about 10 to the minus 2. You use vinegar. Vinegar has a pH that is 3, which means it is acidic. The concentration is 10 to the minus 3. Coffee, a pH about around 5. Okay, this is an acidic drink. The concentration 10 to the minus 5. So you can go on like that. There is only one substance that is absolutely neutral. It's pure water. There is nothing that is more pure than water. So water is the only substance, when it's exclusively water in a bucket or in a glass, where you are going to have a pH that is equal 7. Anything else is going to have a pH that is either above 7 or below 7, right? Now we can make solutions and do design whatever pH we want. In the basic end of the spectrum, seawater, for instance, has a pH that is around 8. This is basic. Baking soda that you commonly use has a pH that is around 9. Okay, just to give you an idea. Now, pH is going to be very important in living systems because our body is going to have many chemical reactions that need to run. And these chemical reactions are working in an optimal environment. Now, most of the chemical reactions that occur in our system require enzymes. Enzymes, which are proteins, are going to be functional optimally at a certain pH. If you consider, for instance, the stomach is very acidic, extremely acidic, actually about 2. Now, there are some enzymes that work in the stomach, but this enzyme cannot work in the mouth because the mouth is not acidic. All right? This enzyme requires an acidic environment. Likewise, the enzymes that are found in the small intestine work in a slightly neutral to slightly basic pH. Compared to the stomach, this enzyme can never work in the stomach. So it becomes very important to maintain a balance in the pH based on the place in the body. Now, having said all of this, let's take a look at the molecules of life. Now, we talked about how atoms come together to form molecules. You, as individual, you are made up of bunch of molecules, some of them being very large. Now, you notice that between the time you were a baby and now you are most likely adult or late teen, you have grown. The question is, what happened so that you can grow? Well, your body has synthesized parts that cause your body to grow, right? Your limbs have grown in size. Your head has grown in size. All your entire person has grown in size since your childhood, right? You have a number of chemical reactions that occur. These chemical reactions are really of two kinds. One is the reactions of synthesis. These reactions of synthesis are going to allow the building of large blocks so that you can grow. And you have reactions of breaking down. These reactions of breaking down are going to cause rearrangement of the very molecule you ingest when you eat, right? So, these reactions in synthesis and reactions of breaking down fall into two categories. One is the dehydration reaction. This dehydration reaction allows to bring two small subunits together by simply removing water. We are going to see the detail in a minute. By removing water, the cell, through dehydration synthesis, is able to build large molecules. While hydrolysis, which requires input of water, is going to cause a breakdown. And that breakdown is essential for your growth and for your maintenance. Know that in the body, these two reactions go hand in hand. One doesn't go without another. Think a minute. If your body can only and exclusively do dehydration reaction, what happens is that your cells are going to take up volume non-stop. That is a problem. Likewise, if your cells can only do breaking down, your cells are going to lose volume so that at the end of the day, well, the cell is going to die. So either way, one reaction doesn't go without the other reaction. So here, you have a subunit right here that is going to interact with another subunit. Most of the time, these reactions are going to involve enzymes, right? With the involvement of enzyme, a dehydration reaction is going to take place, which means a release of a water molecule. By releasing a water molecule, now we're going to have a larger molecule here with a new covalent bond that exists here that didn't exist before. So that's reactions of synthesis or reactions of building up. Then you have reactions of breaking down hydrolysis. So here you're going to have an input of water to actually right here, the input of water occurs here so that this covalent bond can be broken. That covalent bond cannot be broken in our system without the help of enzymes. So enzymes are going to come and help that so that now we have two subunits that are separated. When you think of digesting the food you ingest, right? Hydrolysis reactions are responsible for that digestion. Now, we've talked about the reactions of building up and the reactions of breaking down. Let's address the molecules of life, because these reactions of building up and breaking down, the dehydration synthesis reaction and the hydrolysis reactions, are responsible for the production of complex molecules. Among the complex molecules of life, we are going to have four categories. One is the carbohydrates or sugars. Two is the lipids or fats and oils. Then we're going to have proteins. Some of them are going to be structural proteins. Others are going to be functional. Among the structural proteins, you have, for instance, the proteins that make up your hair, the proteins that make up your nails, right? Among the functional proteins, you have all the enzymes that break down the food you ingest. And then you have the nucleic acids. Among the nucleic acid, we're going to have DNA and another molecule called RNA. DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. At this point, we are going to dive into each of the categories of molecules. What is their structure? What are they for in our body? So the first step is going to be the carbohydrates. Remember, carbohydrates are also known as sugars, right? Carbohydrates are made up of building blocks. These building blocks, we call them monosaccharides. These monosaccharides are made up of elements including carbon, hydrogen, oxygen. These are the most common elements that are found in sugar. In general, these elements are present in a proportion that is 2:1. In other words, for one oxygen and one hydrogen, for one oxygen and one carbon, you are going to have two hydrogen. Let me repeat that. For one oxygen and one carbon, you are going to have two hydrogen. We are going to see that detail in a minute. Carbohydrates or sugars, they function in our body in two ways. One, they play the role of source of energy, whether you're talking about short-term source of energy or long-term source of energy. They are also going to play a very important role in the structure of our body. Then we are going to find beyond the monosaccharide very complex sugars. Okay? These complex sugars are basically what we call polysaccharides. Mono means one, poly means several, right? Several building blocks. So, what are some examples of monosaccharides? Some examples of monosaccharides. Here, we are going to have a molecule with 6 carbon atoms, 6 oxygen atoms, and 12 hydrogen atoms. An example of monosaccharide is going to be glucose. Glucose is C6H12O6. I want you to remember that. Another molecule that has the exact same formula is galactose. Galactose, it's another molecule, monosaccharide, that is C6H12O6. A third one I would like to mention, if you allow me to, it's going to be mannose. Mannose. A fourth one is fructose. Fructose is found in fruits. Glucose is found about everywhere. Galactose is found in milk. Just to give you a reference. So these are simple sugars. Now these simple sugars, through dehydration reaction, these two guys can react together to form a disaccharide. D means two, saccharides means sugar. A two-sugar molecule by which you have a covalent bond that develops between the two sugars, right? Notice that here you have 12 carbon atoms, 22 hydrogen, and 11 atoms of oxygen. This is so because you lost one oxygen here and you lost 2 hydrogens here. It's very important as you want to determine the overall chemical formula of a complex molecule to consider the fact that you are going to have a dehydration reaction, a dehydration during the reaction. There we go. Okay? So I can ask you, well, if you have 10 molecules of glucose, you want to bring together through a dehydration reaction, what do you think is going to be the final formula, the chemical formula of that molecule? Okay? Think about that. Now, we are going to go, oh, by the way, I didn't say this. A glucose and a glucose molecule form a molecule called maltose. There are other disaccharide I want you to remember. One other example of disaccharide is lactose. That is found in milk. And lactose, it's basically a glucose molecule to which a galactose molecule is attached. I want you to remember that because you drink milk on a regular basis. Another example I want you to remember, it's that of sucrose. Sucrose is table sugar. Table sugar is simply glucose to which fructose is attached. This is another example of disaccharide. So please remember 3 examples of disaccharide. We have maltose, glucose and glucose. We have lactose, glucose and galactose. And we have sucrose, glucose and fructose. I want you to remember these examples because these are things that you are going to encounter on a regular basis in your life. Now, the polysaccharides. The polysaccharides are going to be very complex molecules, gigantic molecules, right? This polysaccharide occur in many forms in our environment and in us. We store glucose in a form of glycogen. When I say we human, but really what I should say, it's animals. Animals store glucose in the form of glycogen, while plants store glucose in the form of starch. Okay, so these are 2 examples of polysaccharide, glycogen and starch. Now there is another example of polysaccharide I want you to remember, it's cellulose. Cellulose is found as a structural component of a plant cell wall. I want you to remember that. Cellulose is a polysaccharide. Now let's take a look at lipids. One thing that is characteristic of lipids is that they don't know how to mingle with water. They do not dissolve in water. I want you to remember that while saccharides know how to dissolve in water, they are used as an energy source, long-term energy source. Very importantly, they are found making up cell membranes. In the body, they are going to be found as fats and oils, but also as phospholipids and steroids. We are going to address that. So the fats, in general, they are going to be found abundantly in animals. They tend to be solid at room temperature. They are used as long-term source of energy. They are also going to be used for insulation. You've noticed, for instance, that if you consider, as we are in the winter, and you consider deers out there, if you kill a deer and you cut the skin of the deer, you're going to notice that underneath there is a thick layer of lipid. And that layer is thicker in the winter compared to the summer because that lipid layer is going to be used as insulation for the deer, right? It's also going to be, the fat is also going to be used as a cushion for organs such as the heart. The oils in general are going to be found abundant in plants. They are typically liquid at room temperature, Now, the most common lipids that we are going to find in the human body are going to be what we call triglycerides. Triglycerides. What are triglycerides? Triglycerides are simply one glycerol molecule to which 3 fatty acids molecules are attached. Through dehydration reaction, we are going to have here a fat molecule or a triglyceride molecule. Now, some fats are going to be saturated, other fats are going to be unsaturated. Fats that are saturated will likely be solid at room temperature, while fats that are unsaturated are going to be likely fluid at room temperature. And by the way, that is the reason why oils are fluid at room temperature. They are fluid at room temperature because oils contain more unsaturated fat compared to saturated fatty acid, while animal fat is going to be solid at room temperature because animal fat has a high amount of saturated lipid versus unsaturated lipid. Now, what is really the difference when we talk about saturation, unsaturation? Well, when you look at this fatty acid right here, this fatty acid right here, between from one carbon to another, they are connected by single covalent bonds. And every single of this carbon here has hydrogens attached. So you can see hydrogens that are attached to these carbons. Therefore, these carbons are saturated with hydrogen. But if you consider this fatty acid right here, this fatty acid, while it has simple covalent bonds between carbon, but there is one particular area here where we do see a double bond. That double bond between these two carbon is going to cause some hydrogen to go away. Now, this fatty acid now is unsaturated because it doesn't have the maximum possible hydrogen, right? Because of the double bond here. That's one. Two, it's the fact that this double bond is going to cause the formation of a kink. That kink is the reason why these lipids are going to have a lower melting point compared to the same kind of lipid that would be saturated. Let me repeat that. The presence of this double bond is going to cause a kink in the molecule, the structure of the molecule. That kink is going to be the reason why the melting point of that molecule is going to be lower than the melting point of a similar molecule that does not have a double bond. So we talked about the fats. Oftentimes, these fats are going to be modified in our cells. How so? They are going to be modified so that one of the fatty acid is going to be replaced by a phosphate group, so that we are going to have a fat molecule that is phosphorylated. We call this fat molecule that is phosphorylated phospholipid. That phospholipid is going to be made up of a glycerol molecule, two fatty acids, 1 and 2, and a phosphate head. That phosphate head is very much polar, so that we are going to have a polar head that knows how to interact with water, and a hydrophobic tail that is non-polar. It's not going to mingle with water. Now, if you consider that cells are in a system that is mainly dominated by the presence of water, these phospholipids in presence of water are going to spontaneously form what we call a phospholipid bilayer. So you have one layer here of phospholipid, and we have another layer here of phospholipid. So this is a phospholipid bilayer. Bilayer. Okay? Now, this phospholipid bilayer is organized such that you're going to have an outside of the cell and an inside of the cell. In the outside of the cell, you're going to have a bunch of water. In the inside of the cell, you're going to have a bunch of water. In between, you're not going to have water because everything here is not mingling with water. So the phospholipids are part of biomembranes and are going to be important that way. The steroids, a very popular steroids that I want you to know of is cholesterol. We have a lot of cholesterol in our body. Cholesterol can be used as raw material for producing other steroids, including testosterone in males and estrogen in females. These are two hormones, right? Testosterone is a hormone that is abundant in males, while estrogen is abundant in females. Now, let's take a look at proteins. Proteins are going to be made up of subunit building blocks that are called amino acids. Proteins play a very important role in our body because they are going to play different functions, including some of them being hormones, others being enzymes, others are antibodies, others are going to be responsible for the transport of chemicals in the body. Now, one thing that is very important for you to remember about proteins is that these molecules are very sensitive to temperature change. They are sensitive to temperature change, they are sensitive to pH change, they are sensitive to radiation. So any extreme change is going to cause a loss in the shape and the function of a protein. We call this denaturation. Again, denaturation is the loss of shape, one, and function in a protein. That loss of shape and function in a protein can be caused by a drastic change in the temperature, a drastic change in the pH, or a drastic change in radiation in the environment. This is the reason why it is very important for our body to keep the inside temperature at 37 degrees Celsius. That's why we have homeostasis, right? It is very important also to maintain our internal pH at around 7.4 for the same reasons. Now, what do amino acids look like? Well, amino acids are going to be characterized by the fact that they all, absolutely all, going to have what we call an acid group here, a carboxyl group here, and an amino group here. All amino acids are going to have a carboxyl group and an amino group, regardless of the amino acid. However, these amino acids are going to be different one from another by a side chain here that is different. For valine, the side chain is as you see here. For glutamic acid, the side chain is what you see here. For lysine, the side chain is what you see here. So that we have 20 different kind of amino acids. Technically 21, but I want you to remember 20. Okay? I'm not going to ask you to remember the name of the different amino acids, right? This is human biology. It's not biochemistry. So I'm not going to ask you to remember the name of this amino acids. What I want you to remember is that the amino acid have functional groups including carboxylic function and amino group. This carboxylic function and amino group are what is going to allow amino acids to come together through dehydration synthesis to form large molecules or proteins. Now, the proteins, as they are synthesized, they are going to go through levels of folding. The first level of structure of a protein is going to be the primary structure. That primary structure, it's nothing but the linear order of amino acids after synthesis of the protein. But we do know that amino acids, some of them are polar, some of them are charged, some of them are non-charged. Because of that, these amino acids will start interacting with each other so that they are going to adopt a secondary structure. That secondary structure is basically a 2D shape. a 2D shape that is caused by folding because of hydrogen bonds that develop because of some interactions that develop between amino acids. Beyond that secondary shape, you are going to have the tertiary structure. The tertiary structure or 3D shape is the single most important characteristic for the function of a protein. Let me repeat that. The 3D shape is the single most important characteristic for the function of a protein. In other words, regardless of the sequence of a protein, if a protein loses its 3D shape, most likely it is going to lose its function. Remember, we call this denaturation, right? Then you have a quaternary structure. In the quaternary structure, two perfectly folded proteins in their tertiary structures are going to come together to form a functional unit. An example of that is, for instance, hemoglobin. One unit of hemoglobin protein is not enough to carry oxygen. Two are not enough to carry oxygen. Even three are not enough. You need 4 molecules of hemoglobins to come together through some hydrogen bond relationship to actually create a functional unit. Otherwise, there is no functional unit. Okay, so here again, we have the primary structural level of organization of proteins. Then we have the secondary structural level. And by the way, in the secondary structural level, there are two kinds of secondary structural level. We have one that we call the alpha helix, in which the folding is going to happen like a spring, right? And that spring is stabilized by hydrogen bonds. I want you to remember that. The second type of secondary structure of protein is going to be the beta-pleated sheets. These beta-pleated sheets are going to be like an accordion. And this accordion are sheets that are stabilized by hydrogen bonds. I want you to remember that. Then, the tertiary structure, which is the 3D, it is also stabilized by hydrogen bonds. So here in secondary structure, we have hydrogen bonds that are important. In tertiary structure, we also have hydrogen bonds. that are also important here. And in the quaternary structure, we also have hydrogen bonds that are important here. Please remember all of this. In the primary structure, hydrogen bonds do not play a role, only covalent bonds. And by the way, when you bring two amino acids together to form a new bond, that covalent bond carries a specific name. It's called a peptide bond. Peptide bond. So that when you have one amino acid to which another amino acid is going to be connected through dehydration reaction, therefore a peptide bond forming, we have what we call a dipeptide. D means two. Dipeptide. If we have three amino acids connected together, we're going to have a tripeptide. If we have so many, then we are dealing with a polypeptide. Polypeptide, just like polysaccharide, right? In this case, polypeptide, a protein is a polypeptide. I want you to remember that. Now, the nucleic acids. What are nucleic acids? Nucleic acids are made up of building blocks that are called nucleotides. I want you to remember that. These nucleotides come together and form a sequence. That sequence is what is going to determine the DNA you find in plant cells and the DNA you find in human cells. We know that these two DNA are different. Obviously, plants are stiff while animals are not, right? And this is because of the information in the DNA. Anyway, let's close that parenthesis. Now, DNA, the nucleic acid, is going to contain the information necessary for proteins to exist. Without that information, forget about producing proteins, right? Nucleic acid include RNA and DNA. We are going to go in the details here. Now, the building blocks of nucleic acids that we call actually nucleotide are made up of three parts. One is the phosphate group. Two, it's a 5-carbon sugar. Three, it's a nitrogenous base. The 5-carbon sugar occur in two kinds. We have one that is called ribose. That is the 5-carbon sugar. So 5-carbon sugar that is found in RNA. While the 5-carbon sugar found in DNA is called deoxyribose. I want you to remember that. Deoxyribose in DNA, ribose in RNA. Now, the nitrogenous bases occur in five kinds. One is adenine, always abbreviated A. Two, it's guanine, also abbreviated G. Adenine and guanine are double-ringed purine. These are some kind of nitrogenous compound. Then we're going to have cytosine abbreviated C and thymine abbreviated T. These two, cytosine and thymine, are single-ring pyrimidines. In addition to cytosine and thymine, though, you are going to have uracil, abbreviated U. Now, as we look at DNA versus RNA, DNA is going to comprise adenine, guanine, cytosine, and thymine, where DNA is a double-stranded molecule in which A always pairs with T, while G always pairs with C. On the other hand, though, RNA, which is a single-stranded molecule, is going to have adenine, guanine, cytosine. In place of thymine, RNA is going to have uracil, right? Let me show you here. So here we have DNA, which is a double-stranded molecule, right? That double-stranded molecule has nucleotides, and this nucleotide have a phosphate group right here, a 5-carbon sugar, deoxyribose, and a nitrogenous base, right? That nitrogenous base can be adenine, can be guanine, can be cytosine, can be thymine. Notice that adenine pairs with thymine, guanine pairs with cytosine, and these two strands are stabilized by hydrogen bonds, as you can see here, right? Between adenine and thymine, you have 2 hydrogen bonds, while between guanine and cytosine, we have 3 hydrogen bonds. But don't worry about that. I'm not going to bug you with how many hydrogen bonds exist between adenine and thymine and between guanine and cytosine. That's not important. What is important is that the double-stranded helix molecule of DNA is stabilized by hydrogen bonds. You have to know that. Another thing you have to know, that adenine pairs with thymine while guanine pairs with cytosine. You have to know that. DNA, it's a double-stranded molecule. RNA, on the other hand, it's a single-stranded molecule, always, right? That RNA actually is going to have nucleotides with a phosphate group, a 5-carbon sugar, and a nitrogenous base. There are 4 kinds of nitrogenous base occurring in RNA. We're going to have guanine, we're going to have uracil, we're going to have adenine, and cytosine. So anytime you see a molecule of nucleic acid containing uracil, it has to be an RNA molecule. If a nucleic acid molecule contains thymine, it has to be DNA. There is no such thing as a molecule of nucleic acid that has both DNA, that has both thymine and uracil. That does not exist, at least as far as we know. Now, let's run an overall comparison. An overall comparison between DNA and RNA. DNA contains as 5-carbon sugar a deoxyribose, while RNA contains a ribose. The nitrogenous bases found in DNA are adenine, thymine, cytosine, and guanine, while in RNA we have adenine, uracil, cytosine, and guanine. DNA, it's a double-stranded molecule, while RNA, it's a single-stranded molecule. Finally, let's summarize all the molecules of life, the building blocks, and their functions. Let's start with the carbohydrates. Carbohydrates, some examples of carbohydrates are going to be monosaccharides, disaccharides, and polysaccharides. Please go over the lecture to have some examples of monosaccharides, some examples of disaccharide, and some examples of polysaccharide. We did cover those. The building blocks are going to be simple sugars, in this case glucose. What is their function? They are going to be important for energy source. They are also going to play a structural role. The lipids. The lipids are going to be fats, oils, phospholipid, and steroids. The building blocks of lipids are going to be glycerol and fatty acids. Lipids are going to be used as source of energy, long-term source of energy. They are going to play a very important role in forming membranes. They are also going to play a very important role in making hormones. Please remember that. Hormones. Testosterone, estrogen, to name only these two. The proteins, they are going to be structural and functional, like enzymes. Some are going to be carriers, transporters. Some are going to be hormones. Some are going to allow contraction, movement, right? The building blocks are amino acids with functional group being amino groups and acid groups here, right? What is the job of